I'd like to thank the organizers for giving me the opportunity to, giving me the opportunity to share with you a little bit about RGD's data and tools for translational research in Otitis Media. And I would like to mention that I do not have any disclosures.
You want to take the pointer? I think you have to set it. All right.
I don't care either way.
Yeah, we can do it here. Yes, I guess you have to. Okay, now you have the light. Okay.
Yes. Okay. And I'm hoping that this will work. Yeah. And make me do it differently than I do in my rehearsals.
I'm sorry.
Okay. RGD is a multi-species resource for translational research and comparative genomics. We house data for 10 mammalian species, including human and chinchilla. And just for those who use mouse and rat. We integrate data derived from expert literature curation with data imported from other sources. We provide an extensive and well-rounded corpus of genetic, genomic, disease, and physiological data. And we provide innovative tools for data analysis. Our data and tools are accessible from the menu bar at the top of most of the RGD pages. Data can also be accessed using the general search box or these targeted searches. Our tools can be accessed using the analysis and visualization links on the front page. And the species icon link that are showed on the front page link to informational landing pages for each species. Our species portals provide easy access to species-oriented information, data, and tools. For instance, this is the page for the human species. The layout is essentially the same for all of the species portals, although depending on the data and tools available for each, there are differences. So this is the top of the page for chinchilla. The top of the page gives information about chinchilla in this case and other species that are used as models of human disease. Below that, there is information and links to chinchilla data, to RGD tools with chinchilla already chosen as the species, to links to genome and gene resources at RGD, NCBI, and Ensemble. And all of these links are also available in the right sidebar, along with links to chinchilla research laboratories and selected publications. Hello. There we go. Wait. Okay. So I don't have time to go through everything that's shown on this page, but I would like to highlight a couple of things. And the first thing that I would like to highlight is the Chinchilla Phenominer. Phenominer is a data mining tool for quantitative phenotype measurement data for both rat and chinchilla. The data in Phenominer is a combination of curated data and direct submissions from researchers. When you first access the page, got myself in advance. Yes, the link from the Chinchilla portal directly accesses the Chinchilla Phenominer. In the Phenominer, selecting terms in the bottom section adds them to the corresponding bins in the top section. Data can be chosen on the basis of any or all of 4 criteria: the source of the chinchilla, clinical measurements, measurement methods, and experimental conditions. Once selections have been made, click Generate Report to view the associated data. On the results page, there are 3 sections: a graph, a table, and filters in the left sidebar. All 3 of these work together so that filters that are selected on the left will remove bars from the graph and rows from the table, and sorting the table reorders the bars in the graph. Colored by functionality gives you options for coloring the bars on the graph. And here the table is sorted by condition and colored by the sex of the animals. Options are given to download all of the records that match your original search criteria, or just a filtered set if filters have been applied. So switching gears a little bit, RGD's general search searches across data types and across species. So a search of Otitis Media will return genes in each of the species, human variants from NCBI's ClinVar database, and links to vocabularies that RGD uses for functional annotations, including the disease ontology. So if you click that link to the disease ontology, you access a page that shows the 32 disease ontology terms that match the search criteria Otitis Media. RGD uses ontologies for data standardization, and ontology is a structured vocabulary which is often shown as an upside-down tree structure with fewer, more general terms toward the top and larger numbers of more specific terms as you move down the tree. The leaf icon on the search result page takes you to RGD's ontology browser, which gives you easy functionality to go up the tree to the more general terms and down the tree to the more specific terms. The A icon in the search results links to RGD's ontology report. This is the top of the page for the ontology report for Otitis Media. You'll see that it has information about the term, including a definition, a number of synonyms, and links to related terms in other vocabularies and related information in other databases, such as online Mendelian inheritance in man, or OMIM. Where possible, a genome-level view of the genes annotated to the term is shown. For human, RGD has annotations to Otitis Media for both genes and clinical variants, as shown in this view. For Chinchilla, most of the gene annotations are derived from orthologous genes in other species, as indicated by this ISO, or inferred from sequence orthology evidence code. But RGD curators have reviewed the literature and made experimental annotations directly to chinchilla genes wherever possible. RGD's disease portals provide consolidated views of data related to 15 disease categories. They're accessible from this diseases tab in the top menu bar. If you click that, it takes you to this disease portals landing page that shows you all 15 of the portals, and I will use sensory organ disease as my example. When you access the portal, the first thing that you can do is select a data type. Here, disease is the one that is selected. That's the default. And you can also choose a species, and I've chosen human. Disease portals start with the most general term in the category, in this case, sensory system disease, but you can use the embedded ontology browser to browse down the tree to get more specific disease terms. The portal leverages the structure of the ontology to show all of the objects, in this case, all of the genes, annotated to the term and all of its children. As selections are made, this list of genes is updated on the fly. For human, the genome-wide view of gene positions is shown. There is an embedded copy of the multi-ontology enrichment tool, which I'll talk about in just a minute. And links to additional information are provided at the bottom of the page. And this same functionality is available for all species. So if I go back up to the top and choose Chinchilla, when Chinchilla is shown, the list of genes that's shown is all of the genes in Chinchilla annotated to Otitis Media or any of its more specific children. If an ontology is selected in the gene set enrichment section of the page, as I've selected the pathway ontology here, the list of genes is automatically submitted to the embedded MOIT tool for enrichment analysis. In the results, the table shows the list of enriched terms in the pathway ontology, the number of genes in the list of genes that are annotated to each of those terms, the uncorrected and Bonferroni corrected p-values and odds ratio for each term. Not surprisingly, the most enriched terms for the pathway ontology for that list of Otitis Media genes are terms related to interleukin signaling and immune pathways. And the graph shows the number of genes in blue and the p-value for each term in orange. Each gene in the list of genes in the disease portal links to its respective gene report page. So if I click this mucin 5B gene symbol, it will take me to the Chinchilla gene report page for mucin 5B, which in turn links to that gene's position in the JBrowse genome browser. RGD has loaded genes from NCBI and Ensemble and RNA-seq BAM alignments and coverage tracks from 2 projects, uninfected and infected middle ear mucosa from the Medical College of Wisconsin, and 14 tissues from samples provided by Dr. Lauren Bakelitz's group at the Research Institute at Nationwide Children's Hospital. The first set shows that mucin 5B increases in expression from uninfected through days 3 and 17 of strep infection. The second set shows tissue-specific expression for mucin 5B, as shown here. And this display shows the BAM alignments for 2 exons of mucin 5B from one of the uninfected middle ear mucosa samples. So in conclusion, this brief overview has only given you a small subset of the rich set of data and tools that RGD provides for both clinical and translational researchers. If you have questions or are interested in more information, I'm available and I have some contact information here. But I would like to say that we have a couple of questions for the research community. The Chinchilla genome is only assembled to the point of scaffolds, as you can see here in the JBrowse chromosome dropdown, which is sufficient for some applications, but for true comparative genomics or phylogeny or that sort of thing, chromosome level assembly really would be required. And so our questions are, is there interest in improving the Chinchilla genome assembly? And if so, would groups be willing to do additional genome sequencing to support chromosome level assembly? And with that, I would like to say thank you. I'd like to acknowledge the incredible RGD team and our funders and the researchers who contribute data and faithfully use our website and data, and especially Dr. Kirshner and Dr. Hong here at the Medical College, who provided quantitative phenotype and RNA-seq data, and Dr. Bakelitz, who provided RNA-seq data.